In this experiment, we're going to count the number of codons within human DNA. To make our task easier, we'll just be looking at chromosome 1. There are 22 chromosomes in human DNA, and we're just looking at chromosome 1. What we're going to be doing is counting the frequency of each of the 64 different codons. So there are 64 different codons in DNA, and we're just going to be counting the frequency of each of them. The aim is to see if there is any mathematical relationship between the frequency of one codon and the frequency of another. So let's click start and see what happens. What's happening now is the software is looping through chromosome 1. The number here represents the number of letters that the software has read. This is simply the number of the chromosome that is being read. Here you can see a list of 32 of the codons, and here are the other 32. I've arranged the codons so that the codons in this column are the complement of the codons in this column. Now, as we run the program, the frequency of each codon is shown next to it, and here. And the ratio of the frequency of this codon to this codon is shown here, and of this codon to this codon is shown here, and of this codon to this codon is shown here, and so on. So this column here represents the ratio of these frequencies to these frequencies. What you might notice already is that there does seem to be a mathematical relationship between a codon and its complement. It looks like the frequency of any codon in this column matches the frequency of its complement in this column. This is a very strange phenomenon. After all, DNA codes for very complex organisms. The last thing we would expect is that the letters which comprise that DNA form this balanced mathematical pattern. Anyway, we've now looped through almost 1 million letters. There are 3 million letters in chromosome 1 altogether, so it will take another couple of minutes. Once we have gathered all the finished looping through the chromosome, then we're just going to enter the frequencies of the codons and their complements into an Excel graph and get a visual on the relationship between the frequency of codons and the frequency of their complements. The graph will show more clearly the mathematical relationship between these two. What you're looking at is the beginning of something really big. This pattern just should not be here. By all the laws of random chance and evolution, this pattern shouldn't be here at all. Okay, we're about halfway through. Sometimes you'll see the program actually stops, and this is because not all of the sequence of DNA is currently known. There are some large areas within each of the chromosomes where we just do not know whether it is an A, a C, a T or a G, in which case normally it's shown as by the letter N, and once the software loops past the N regions, which basically stands for unknown, then it just carries on counting the number of codons. What is also apparent from this is that this mathematical equality between the frequency of a codon and its complement occurs right the way through a chromosome. It's not as if one end of a chromosome is a mirror of the other half. It's a consistent equality all the way through. So right from the beginning, when I started this program, the ratio was already 1 or 0.99, and it wasn't as if the half of the DNA mirrors the latter half of the DNA mirrors the first half. It's more like the mirror is, as it were, dispersed right the way through the DNA. I find this phenomenon intriguing. It's almost as if our DNA contains a mirror of itself, that we were made in the image or in an image of something, because our DNA contains an image of itself. Who would have guessed that our DNA contains this perfect mathematical balance between codons and complementary codons. The occurrence of the codons doesn't seem random at all. As you can see, the different codons all occur with different frequencies. It's now stopped because it's finished analysing chromosome 1. And as you can see, looking down the list, each of these codons occurs with a different frequency. Some a lot more than others. Now compare this list to the complements: 28, 28, 15, 15, 15, 15, 14, 14, 17, 17, 12, 12, 15, 14.9, 14.9, 17, 17, 15, 15, 13, 13, 9, 9, 16, 16. There appears to be a strong correlation between the two columns. Obviously, we can apply a correlation test to this data to get a precise mathematical measure of the strength of the correlation, but it looks extremely strong to me. What we're going to do now is take the numbers here, just copy and paste them, Control C, and we're going to paste them into Excel. So we just paste them in, and now what we're going to do is simply look at a graph, insert a graph, line and line. And from this graph, I think you'll agree that we have almost like a fingerprint identity between the frequency of 32 of the codons and the frequency of their complement. In fact, if we place these two lines on top of each other, you'll see that the fit is almost perfect. That's because one line is right on top of the other one. So we can conclude that there is definitely a strong relationship between the frequency of any codon and the frequency of its complement. It's a mathematical relationship of equality, 1 to 1.